Dear viewers, this video is part 1 in the 4-part series on cancer cell biology in the Medicine for the Common Man series. Cancer cell biology. Cancers are characterized by unregulated cell division, avoidance of cell death, tissue invasion, and the ability to spread to other areas of the body, that is, metastasize. A neoplasm is benign when it grows in an unregulated fashion without tissue invasion or metastasis. The presence of unregulated growth, tissue invasion, and the ability to metastasize is characteristic of malignant neoplasms. Cancers are named based on their origin. Those derived from epithelial tissue are called carcinomas. Those derived from mesenchymal tissue, that is connective tissue, are sarcomas. And those derived from hematopoietic tissues are leukemias, lymphomas, and plasma cell dyscrasias, including multiple myeloma. Note: leukemia is cancer of the body's blood-forming tissues, including the bone marrow and the lymphatic system. Many types of leukemia exist. Some forms of leukemia are more common in children. Other forms of leukemia occur mostly in adults. Leukemia usually involves the white blood cells. Lymphoma is a cancer of the lymphatic system, which is part of the body's germ fighting network. The lymphatic system includes the lymph nodes, that is, lymph glands, spleen, thymus gland, and bone marrow. Lymphoma can affect all those areas as well as other organs throughout the body. Plasma cell dyscrasias form a heterogeneous group of diseases characterized by the expansion of the number of monoclonal bone marrow plasma cells that produce monoclonal immunoglobulins. The incidence of these diseases is as high as 5% in adult individuals. Cancers nearly always arise as a consequence of genetic alterations, the vast majority of which begin in a single cell and therefore are monoclonal in origin. However, because a wide variety of genetic and epigenetic changes can occur in different cells within malignant tumors over time, most cancers are characterized by marked heterogeneity in the populations of cells. Dear viewers, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Variable extrinsic factors in the cancer environment. In addition, extrinsic factors in the cancer environment, example, the stroma, infiltrating cells, various cell-to-cell interactions, spatial orientation, secreted factors, and availability of oxygen and nutrients vary in different areas within the tumor or different metastases, compounding this heterogeneity. This heterogeneity significantly complicates the treatment of most cancers because it is likely that there are subsets of cells that will be resistant to therapy for a variety of reasons and will therefore survive and proliferate even if the majority of cells are killed. A few cancers appear to, at least initially, be primarily driven by an alteration in a dominant gene that produces uncontrolled cell proliferation. Note: dominant refers to the relationship between two versions of a gene. Individuals receive two versions of each gene, known as alleles, from each parent. If the alleles of a gene are different, one allele will be expressed, that is the dominant gene. An allele is a variant form of a gene. Some genes have a variety of different forms, which are located at the same position or genetic locus on a chromosome. Humans are called diploid organisms because they have two alleles at each genetic locus, with one allele inherited from each parent. Examples include 1. Chronic myeloid leukemia. Note: chronic myeloid leukemia (CML) cells contain an abnormal gene BCR-ABL that is not found in normal cells. This gene makes a protein BCR-ABL which causes CML cells to grow and reproduce out of control. BCR-ABL is a type of protein known as a tyrosine kinase. The Philadelphia chromosome, PH chromosome, that is the BCR-ABL fusion gene, is formed by fusion of the sequences from ABL1, that is Abelson gene, at 9q34 to the BCR breakpoint cluster region gene sequences at 22q11. T922q34q11, that is the notation for this chromosome translocation. The translocation between chromosome 9 to 22 and chromosome at location 34 from the long arm Q of the chromosome 9 to the long arm of chromosome 22 at 11 position. 2. About half of melanomas. The gene BRAF. BRAF is a serine threonine protein kinase activating the MAP kinase ERK signaling pathway. About 50% of melanomas harbors activating BRAF mutations. BRAF has been implicated in different mechanisms underlying melanoma genesis. 3. Burkitt lymphoma (BL) is a highly aggressive B-cell neoplasm characterized by the translocation and deregulation of the MYC gene on chromosome 8. BL is derived from germinal center B cells. It is thought to arise from B cells. BL development depends on the expression of MYC gene, which encodes for the CMYC protein transcription factor located on chromosome 8q24 and regulates cell proliferation, differentiation, and apoptosis. BL is characterized by inappropriately high levels of CMYC, most commonly due to translocation of the long arm of chromosome 8 containing the MYC gene and the IG heavy chain gene on chromosome 14. CMYC overexpression leads to rapid B cell proliferation, accounting for the rapid doubling time of BL tumor cells. Genes that can promote cell growth, when altered, are often called oncogenes. Viral genes have normal counterparts with important functions in the cell and had been captured and mutated by viruses as they passed from host to host. However, most human cancers are characterized by a multiple step process involving many genetic abnormalities, each of which contributes to the loss of control of cell proliferation and differentiation and the acquisition of capabilities such as tissue invasion, the ability to metastasize, and angiogenesis, that is, the development of new blood vessels required for tumor growth. These properties are not found in the normal adult cell from which the tumor is derived. Indeed, normal cells have a large number of safeguards against DNA damage, including multiple DNA repair and extensive DNA damage response mechanisms, uncontrolled proliferation, and invasion. This figure shows the different stepwise progression of colon carcinoma. First, carcinogen induced mutations initially, then additional driver mutations, later on additional mutations with emergence of subclones aiding invasion and metastasis. Many cancers go through recognizable steps of progressively more abnormal phenotypes: hyperplasia, to adenoma, to dysplasia, to carcinoma in situ, to invasive cancer with the ability to metastasize. See figure 1.1 shown earlier. For most cancers, these changes occur over a prolonged period of time, usually many years. Characters of malignant cells. 1. Deregulated cell proliferation. Loss of function of inhibitory growth factors. Tumor suppressor genes like p53. Increased action of positive growth regulators. Oncogenes like MYC leads to aberrant cell cycle control, including loss of normal checkpoint controls. 2. Failure to differentiate. The stem cells are cells which have the capacity to develop into different kinds of tissues and specialize in their functions, such as cartilage, muscle, and nerve tissue. The cancer cells lose their tissue differentiation and specialization and fail to differentiate. Many retain their stem cell properties. Example, in leukemias, the translocations of chromosomes leads to production of gene products that suppress differentiation pathways. 3. Loss of normal apoptotic pathways like inactivation of apoptotic p53 pathway and increase in anti-apoptotic BCL2 family. This enhances occurrence of chromosomal defects and multiplication of such chromosomally defective cells. 4. Defects in chromosomal repair pathways, leading to single or multiple nucleotide defects, leading to microsatellite instability, that is MIN, or more commonly chromosomal instability, that is SIN, leading to aneuploidy, that is increase or decrease in chromosomal number. This is due to defects like loss of function of p53, BRCA1/2 protein, mismatch repair genes, DNA repair enzymes, and the spindle checkpoint. See the figure 1.1 earlier shown. 5. Loss of replicative senescence. Normal cells stop dividing after 20 to 50 cell doublings. Multiplication is controlled by p56 and p53 pathways. The normal doublings lead to telomere loss, leading to death of cells. Cells that survive often harbor gross chromosomal abnormalities and the ability to continue to proliferate. These cells express telomerase, which maintains telomeres and is important for ongoing growth of these cells. Many human cancers express telomerase. 6. Non-responsiveness to external growth inhibiting signals. Cancer cells have lost responsiveness to signals normally present to stop proliferating when they have overgrown the niche normally occupied by the organ from which they are derived. Our understanding about this mechanism of growth regulation remains limited. 7. Increased angiogenesis. Due to increased gene expression of pro-angiogenic factors like VEGF, FGF, IL-8, angiopoietin by tumor or stromal cells, or loss of negative regulators, endostatin, tumstatin, thrombospondin, etc. Note, VEGF, vascular endothelial growth factor, is a potent angiogenic factor and was first described as an essential growth factor for vascular endothelial cells. VEGF is upregulated in many tumors. FGF, fibroblast growth factors, are potent regulators of cell proliferation and differentiation. They are critically important in normal development, tissue maintenance, wound repair, and angiogenesis in somatic stem cells. IL-8, interleukin-8, is a chemoattractant cytokine produced by a variety of tissue and blood cells. It has a distinct target, specificity for the neutrophil with only weak effects on other blood cells. Interleukin 8 attracts and activates neutrophils, that is a type of white blood cell in inflammatory regions. Angiopoietins. Angiopoietin are peptides involved in vascular development. Endostatin induces microvascular endothelial cell apoptosis and inhibits endothelial proliferation and angiogenesis, which may result in a reduction in tumor burden. Tumstatin. Tumstatin is a protein fragment of collagen that serves as both an anti-angiogenic and pro-apoptotic agent. Thrombospondin. Thrombospondins are a family of with anti-angiogenic functions. The thrombospondins regulate migration, cellular attachment, and invasion. They are secreted in the extracellular matrix where they interact with other growth factors and signaling molecules to influence anti-angiogenic and anti-proliferative effects. 8. Cell mobility and ability to move through extracellular matrix and into the other tissues or organs. Loss of cell-to-cell contacts, that is gap junctions, cadherins, and increased production of matrix and matrix metalloproteinases, MMPs, can take the form of epithelial to mesenchymal intertransition, with anchored epithelial cells becoming more like motile fibroblasts. 9. Metastasis. Spread of tumor cells to lymph nodes or distant tissue sites, limited by the ability of tumor cells to migrate out of initial site and to survive in a foreign environment, including evading the immune system. 10. Evasion of the immune system. Downregulation of MHC class 1 and 2 molecules, induction of T cell tolerance, make T cells unable to recognize the abnormal cancer cell, inhibition of normal dendritic cell and or T cell function. Note: MHC class molecules, MHC1 and 2, are cell surface recognition elements expressed on virtually all somatic cells. These molecules signal the cell's physiological state to cells of the immune system, both T lymphocytes and natural killer cells, NK cells. T cells are part of the immune system and develop from stem cells in the bone marrow. They help protect the body from infection and may help fight cancer, also called T lymphocyte. Dendritic cell, a special type of immune cell found in tissues such as the skin, boosting immune responses by showing antigens on its surface to other cells of the immune system. A dendritic cell is a type of phagocyte and a type of antigen presenting cell, APC. 11. Shift in cell metabolism. Complex changes, including alterations due to tumor stress such as hypoxia and energy generation shifts from oxidative phosphorylation to aerobic glycolysis to generate building blocks for malignant cell production and proliferation. In most organs, only primitive undifferentiated cells are capable of proliferating, and cells lose the capacity to proliferate as they differentiate and acquire functional capabilities. Note: differentiation, that is the process of development during which fetal cells specialize and make diverse tissue structures like cartilage, bone, heart, muscle, etc. The expansion of the primitive stem cells is linked to some functional need in the host through receptors that receive signals from the local environment or through hormonal and other influences delivered by the vascular supply. Note: stem cells are special cells from which all other cells with specialized functions are generated. The signals that keep the primitive cells at rest remain incompletely understood. These signals must be environmental. This is indicated by 1. A regenerating liver stops growing when it has replaced the portion that has been surgically removed after partial hepatectomy, and 2. regenerating bone marrow stops growing when the peripheral blood counts return to normal. Cancer cells clearly have lost responsiveness to such controls and do not recognize when they have overgrown the niche normally occupied by the organ from which they are derived. A better understanding of these mechanisms of growth regulation in the context of organ homeostasis is evolving. So, dear viewers, this is the end of part 1 in the 4-part series on cancer cell biology in the Medicine for Anyone YouTube channel. Thank you for watching the video.